So I may call things to order and go ahead and get the seminar started today. Thank you. So it's my distinct pleasure to introduce today's Jones speaker. So Dr. Mike Vahey is coming to us from the Berkeley, where he is a postdoctoral fellow in the Department of Bioengineering working with Dan Fletcher. Mike has a Burroughs Wellcome Career Award at the Scientific Interface, and this independent funding has been enabling him to do some really fantastic research that leverages genetic engineering, microfluidics, and fluorescence imaging to do a number of things, including kind of a mechanistic study of how influenza virus buds from cells and is transmitted. Mike got his undergraduate degree from Carnegie Mellon, BS in electrical and computer engineering. He then went on to the Massachusetts Institute of Technology, where he received a PhD in electrical engineering and computer science, working with Professor Joel Voldman, and that's where Mike acquired his kind of core expertise in microfluidics and microfluidics engineering. And so with that, please join me in welcoming Dr. Vahey as the Jones speaker today.
All right, can everybody hear me okay? I'm not sure if the microphone is picking me up. Is it? Okay, so thank you, Carl, for that introduction, and thank you all for the chance to come visit Dartmouth today and talk a little bit about some of the work that I've been doing as a postdoc at UC Berkeley. So this presentation is essentially about influenza A virus. But the broader question that I think underlies what I'd like to talk about today is that of how do you build a biological structure, and how does the way that you build such a structure ultimately influence its function? So I would propose that the way of building things in biology that we know the most about would be self-assembly. And one way to think about a self-assembling system is that essentially the final assembled structure is in some sense encoded by the properties of its subunits. So for example, if these subunits are proteins, and we know something about the structure or the characteristics of these proteins, then at least in principle we know something about the types of things that they can build. Another characteristic of self-assembling systems is that a lot of their behavior can be described or understood in terms of equilibrium thermodynamics. And while there are often substantial practical challenges in arriving at such a description, self-assembly is something that we now understand at least well enough that we can start to design self-assembling systems on our own. So the example that I'm showing here is work from David Baker's lab, where essentially they've designed proteins computationally such that when they express and purify these proteins and mix them together in a test tube, they spontaneously assemble into these very regular three-dimensional capsid-like structures. So given that self-assembly provides a very robust and reproducible way to build things essentially out of biological parts, it's not surprising that nature leverages this as well. And while there's many examples of this, the one that I want to focus in on for the purposes of my talk today are viruses, and in particular, enveloped viruses. So enveloped viruses are pathogens that at some stage in their replication cycle bud through the membrane of an infected cell. And in this way, they acquire a lipid envelope, and hence the name. So the particular envelope virus that I'm showing here is herpes simplex. And what I want you to take from this electron micrograph is that you'll notice that the viral particles are very uniform, very homogeneous in their size and shape. And although you can't see it from electron micrograph, presumably they're also fairly uniform in terms of their composition, because at least to some extent these things go hand in hand. So this uniformity is a consequence of the way that the virus assembles through a self-assembly mechanism inside of an infected cell. So while a lot of viruses follow this sort of assembly pathway, what I want to emphasize today is that it's by no means universal. And as a counterexample, I want to introduce the subject of what I've been working on for the past few years and what I'll be talking about today. And that's a different enveloped virus, influenza A virus, or the flu. And so what I hope you can see right away from this image is that even in a genetically uniform population such as the one represented here, there's quite a bit of heterogeneity and diversity in the size and shapes of the particles, from these longer filaments to smaller, more spherical viruses. So one way that we can think about this is that while interactions between viral proteins are certainly constraining the characteristics of the final structures that they're assembling into, there are additional stochastic inputs or inputs from the cell during assembly that are also guiding things. So essentially, I'm interested in trying to ask the question and understand if we can begin to understand this sort of assembly process in the same way that we're beginning to understand self-assembling systems. And if we can then take this one step further to try and understand why one set of viruses might prefer to produce very homogeneous progeny, while others favor a different strategy. Okay, so at this point, I think it's important to note that even though influenza and many other human pathogens are heterogeneous in this way, this doesn't seem to be a particular liability for their infectivity or their virulence. And so just to go through a couple of the numbers from the CDC, each year about 1 in 5 people will catch the flu. This can result in about 500,000 deaths annually. And in the case of a pandemic year, as what occurred in 1918, this number can skyrocket up into the tens or even hundreds of millions. Another thing that I want to emphasize, and that I've hinted at already, is that influenza virus is not alone in this sort of tendency to be morphologically heterogeneous. So other important human pathogens, from retroviruses like HTLV to hepatitis C, and then to Ebola, all show different degrees of heterogeneity, either assembling into spherical particles that are sort of broadly distributed in the diameter, to Ebola, which, similar to influenza, can form long filaments that can even have a branched morphology. So all of these are examples of pathogens that are important threats to human health, and that assemble in a way that is not necessarily characteristic of what you would expect for a stereotyped virus that assembles via self-assembly. So we'd like to understand to what extent this could be an adaptive advantage, or if it's a liability, how the viruses are going about coping with it. So I've mentioned so far in the context of influenza that it assembles into heterogeneous particles and that these particles are still infectious. But what still remains unclear is if and how these things might be linked. So the first indication that there could be a connection came in the 1940s when researchers collected essentially influenza virus from a sick patient. And when they looked at these clinical isolates with an electron microscope, they noticed that in contrast to the more homogeneous, uniform 100 nanometer or so spherical particles that they were accustomed to seeing from virus grown in the lab, the natural isolates had a more heterogeneous filamentous morphology. Over the next several decades, researchers identified a constellation of mutations that can confer this switch in phenotype from spherical to filamentous, or from filamentous back to spherical. But the molecular mechanisms that are at work during this transition remain unclear. Even more recently, researchers have shown that this switch is the result of a reversible, in some sense, evolutionary process. And by that, what I mean is that if you collect virus from nature, so in this case from a guinea pig, it will have the filamentous morphology. If you allow it to grow for a few generations in tissue culture, it will evolve towards a spherical phenotype. But if you then take the lab-adapted strain and put it back into an animal, it will revert back in the other direction. So it will regain the filamentous phenotype that I've been referring to. So this, again, provides some evidence that there are selective pressures that are dependent on the growth environment that are favoring one phenotype versus another. But it still remains unclear exactly what these selective pressures might be. So I don't have an answer to that. I have, by the end of this talk, I hope I'll have made my hypothesis clear. But I don't have a definitive answer just yet. So far I've been talking about morphology, but there are other aspects to heterogeneity in virus assembly as well. And so another one that's particularly important is that of composition. So in the context of influenza virus, I think one of the most important examples of this involves two particular proteins that are abundant both on the surface of the virus and on the surface of infected cells. And these are hemagglutinin, or HA, and neuraminidase, or NA. So if you're familiar with the subtype classifications, so for example, H1N1 or H3N2, the H refers to hemagglutinin and the N refers to the subtype of the neuraminidase associated with that virus. So the interesting thing about these proteins is that on some level they have competing activities. And what I mean by that is that hemagglutinin, among other things, is important for binding to the receptor to initiate infection. So this receptor is sialic acid. It's abundant on the surface of cells. And essentially, this is the means by which the virus attaches to start infection. Neuraminidase, on the other hand, is an enzyme that cleaves sialic acid. So it's a receptor-destroying protein. And so given the fact that HA and NA have these competing activities, it seems reasonable enough on some level to suppose that there could be an optimal balance between them. And this is the type of this is the question that people have approached with the following types of experiments. So for example, if you take a wild-type virus and you mutate hemagglutinin so that it has a higher binding affinity, you'll get a virus that replicates rather poorly, presumably because you've thrown these two activities out of balance in some way. If, however, you allow the virus to grow for a few generations, it will gradually restore a more normal rate of replication by accumulating mutations. And in these experiments, the particular mutations and adaptations that the researchers found were twofold. So in one case, they found that the neuraminidase enzyme essentially evolved a longer stalk. So this put the catalytic part of the molecule further away from the viral membrane so that it could essentially outcompete hemagglutinin to cleave sialic acid before it could bind. And in the other way that the virus was able to restore its regular rate of replication was essentially by lowering the binding affinity of HA. And it did this essentially by inserting a carbohydrate close to the binding pocket, presumably sterically occluding binding and effectively lowering the affinity. So again, these are experiments that provide evidence in support of a notion of there being an optimal balance between HA and NA. But it remains unclear exactly what this means on the level of individual virus particles, where there may not be any precise mechanism for regulating just how much hemagglutinin or just how much neuraminidase is packaged. So these are the types of questions that I set out to address. So just to go over these, we're interested in trying to understand how virus morphology and composition are established. We're interested in trying to understand how these properties vary across a population. And finally, we're also interested in understanding why this might matter. So how does morphology, composition, and their variation across a population influence the ability of a virus to replicate and infect the next cell? So in describing our work to try and answer these questions, I've split this talk into two parts. In the first part, I'd like to talk about what determines the properties of a virus and their variation across a population. In order to do this, I want to describe the development of some new measurement tools that I've been working on that allow us to study size and compositional variation across populations at the level of individual viruses. And then along the way, I'd like to introduce some minimal physical models for where the size and compositional variation might arise. Finally, in the second part of the talk, I want to shift gears a little bit then to talk about what this might mean or what the implications of this might be, and how some of the measurements that we're able to now make can give us new insights into the adaptive advantage of heterogeneity in viral populations. But first I want to focus on this part. Okay, so as I mentioned, we're interested in measuring heterogeneity or variation in the size, shape, and composition of a virus. And so with this as our interest, a natural enough way to go about doing this would be to look at the viruses themselves. So for example, if you could imagine attaching a fluorescent molecule to specific proteins within the virus, this would give you contrast where you could start to resolve differences in viral shape and size. And it would also, if you're able to attach the fluorophores in a specific way, allow you to do compositional measurements. So you could look at the relative levels of different proteins that are incorporated into a virus. So since this is a natural enough way to go about answering this question, I want to talk a little bit about some of the challenges that have traditionally made this difficult. So first of all, we have three central requirements for the system that we're developing. So first of all, we'd like it to be innocuous. So in introducing fluorescent molecules on specific viral proteins, we don't want it to have any substantive effect on what it is we're trying to measure, which is the assembly and organization of a virus. Secondly, we'd like the tags to be endogenous. And by this, I mean we want them to be encoded in the virus's genome, so that every copy of a protein that's being expressed off the native RNA segment encodes a copy of the tag. And on a related note, we'd like it to be quantitative, so that the signal that we're measuring with our fluorescence microscope is essentially proportional to the amount of a protein at a particular place in a particular time. So the first way that you might imagine going about doing this would be one that's been tremendously successful in cell biology for decades now, and that's to use a fluorescent protein like GFP or any of its spectral variants. You could imagine genetically appending these to the proteins that you're interested in and imaging different parts of the virus in that way. Unfortunately, as a general rule, attaching a fluorescent protein that's about 25 kilodaltons in molecular weight to any of the structural proteins of influenza virus strongly inhibits or kills replication. So we need to find a more subtle way to do it. Another way that you could imagine going about doing this would be to, instead of encoding the source of fluorescence in the viral genome, you could compromise a little bit and create a cell line that's expressing versions of the viral proteins with fluorescent proteins. You could then infect these with the virus, and then during the assembly process, proteins that are being constitutively expressed in the cell would presumably be incorporated. Unfortunately, in addition to compromising on our ability to be quantitative, because we now have a dark population of proteins that are unaccounted for in our measurement, we're also limited in the sense that even at substoichiometric levels, the incorporation of fluorescently tagged viral proteins can be inhibitory to assembly and replication. So the third approach that we take and that we ultimately is successful is to build off work that's been done in chemical biology over the past several decades now, which is to use site-specific labeling to attach small molecule fluorophores to each copy of an endogenously expressed viral protein. So in effect, what this means is that instead of using a 25 kilodalton fluorescent protein, we use a 5 to 10 amino acid tag. This tag is small enough that it can be attached either at the N or C terminus of a protein or even on internal loops, so that we have a bit of flexibility in terms of finding places where the tag can be inserted without disrupting the function of the particular protein we're interested in. And then we can use enzymes that recognize these short amino acid sequences to conjugate a fluorescent molecule, or we can use fluorescent molecules that will bind directly to that particular sequence. So now that I've talked a little bit about the overall strategy that we're going to employ, I want to talk now a little bit about what exactly it is that we're going to be looking at. So the influenza A virus genome encodes 10 canonical proteins, and at least 8 of these play an essential role in released virions. So when I talk about looking at the structural proteins of the virus, these are the ones that I mean. So first of all, inserted in the viral membrane, the viral envelope, the lipid membrane that surrounds the virus, there are 3 integral membrane proteins. So these are or at least 3 integral membrane proteins, I should say, of viral origin. These are hemagglutinin, or HA, neuraminidase, or NA, which I mentioned previously as being important for receptor binding and receptor cleavage. There's a third membrane protein, M2, which serves as a proton channel that's important both for viral entry and viral exit. And because these 3 membrane proteins have extracellular domains that are presented on the outside of the virus or on the outside of the infected cell, we can use enzymes and small molecule fluorophores that are membrane impermeable to label them. So we have easy access to the extracellular domains of these proteins. And because a lot of these labeling methods have been developed, we have access to a wide variety of enzymes. So we can design the labeling system so that it has orthogonality. For proteins that are inside of the infected cell or inside of the virus, for example, the matrix protein M1, which lines the inside of the viral membrane, and the viral ribonucleoprotein complex, or VRNP complex, which packages essentially each segment of the 8-segment genome, we can use a dye that was developed in Roger Chen's lab almost 20 years ago and can diffuse across the membrane and bind to motifs that we've engineered into these proteins. So in this way, we have access to looking at multiple structural proteins simultaneously. So with this strategy in mind, we can now go about recovering a virus that's able to replicate in a similar way to a wild-type strain. We can use a reverse genetic system that's been developed for influenza virus in the early 2000s. So essentially, we have 8 plasmids, 1 each for each segment of the genome. We modify these based on the tags that we want to use for that particular protein, inserted at sites that are non-disruptive to replication, as I'll discuss in a little bit later. We can transfect cells with these 8 plasmids. These will separately start to synthesize viral genomic-like RNAs as well as mRNAs that go on to become proteins. These proteins and genomic RNAs will assemble into viral particles, and then they'll replicate in the same way as a naturally occurring influenza virus would. So we can collect these viruses, and if we look at a particular version that's been raised in a spherical background, so one that doesn't favor the filamentous morphology, we see something similar to what we would expect. So this is using negative stain electron microscopy, and you can see particles that are about 100 nanometers in diameter, and if you look closely, you can see the spike proteins HA and NA on the surface of the virus. But the important thing now is that because we've inserted these tags, we now are able to use contrast mechanisms other than negative stain EM, in particular fluorescence, to start looking at things. So one way that we can use this is we can infect a cell. About 6 or so hours after post-infection, we can label proteins as they're beginning to be expressed in the infected cells. And this is sort of a characteristic set of images for 2 different types of viruses. So in one case, we're labeling the nucleoprotein, a marker for the viral genome, hemagglutinin and neuraminidase, the 2 proteins that traffic to the apical or top surface of the cell. Another strain here, we can look at the matrix protein M1, hemagglutinin, and the proton channel M2. Importantly, we can also now measure the replication efficiency of these by infecting cells and collecting virus and measuring the titer. And by this metric, we don't see any significant difference between the engineered fluorescent strains and the parental strains from which they're derived. So this gives us some confidence that we can now use this as a tool to do some of the measurements that I described earlier. And so that's exactly what we're interested in, is now trying to use this as a way to try and generate mechanistic models to help us understand virus assembly. So in addition to being able to use this to look at assembly of new viral particles inside of infected cells, we can also infect cells, collect the virus that they shed, and look at that directly. And so that's kind of what I'm showing here. So this is a typical field of view with a scale bar of about 10 microns here. And you can see these are individual viruses. So this is now in a filamentous background, and you can see that their morphology ranges from filaments that can be 10 to 20 microns in length down to diffraction-limited spots that are on the order of 100 to 200 nanometers. So in this particular image, there are 3 proteins that are labeled. In green is the VRNP complex, again, is the marker of the viral genome. In blue is hemagglutinin, and in red is neuraminidase. If we zoom in on this in a little bit more detail, because a lot of these filamentous virions are above the diffraction limit in terms of their length, we can start to resolve features with subviral resolution. And so in particular, if we zoom in on these particles here, you can see there's a green foci at one end of the filament. And that, based on what's been known from electron microscopy for a few years now, is what we're seeing is the viral genome is this bundle of VRNP complexes that localizes to the distal tip of the virus. So this is the part that is first to emerge as the virus is essentially extruding from an infected cell. So this gives us sort of a fiducial mark now where we can collect the viruses and figure out which side is which in terms of what order it emerged from the cell. We can also use images like this to start to reconstruct length distributions. So at least focusing on viruses that are above the diffraction limit of a light microscope, we can measure how the lengths across the population are distributed. And if we do this, again, focusing in a regime where we're above the diffraction limit, we essentially find that it follows a decaying exponential. So at least in this regime, it has a Poisson distribution. So one model that could describe this type of behavior is if you imagine a virus, so this is kind of the surface of an infected cell, the virus is budding outwards. If it's growing with a constant growth velocity, V growth, that's independent of the current length of the virus, and undergoing scission at a uniform rate, this is one way that you could reconstruct a distribution like this. So we can now go back to experiments looking at the surface of infected cells. And although the contrast here may be rough to see, over the course of about 2 hours in this video, you start to see filamentous particles emerging from the surface of this cell. And by tracking the length of these particles over time, we can get measurements for the growth rate of a virus. So for this one in particular, it corresponds to a rate of about 0.1 microns per minute. If you remember me talking about this protein M1 that lines the shell of the virus, this growth rate would correspond to a rate of about 30 molecules per second of this protein being incorporated. So one feature here is that if we look at this within the resolution that we're able to determine, this growth rate appears to be linear, and it appears to be independent of the length of the virus. So this suggests to us that a material is likely being added from the base of the virus. Otherwise, if it were diffusing to the tip before adding onto the budding virus, we would expect to see the growth rate slow down as it gets longer, and that doesn't seem to be the case. But this also gives us some confidence now that what we're seeing could be described by a model as simple as this one, where the virus just grows until scission takes place at some time, where any particular interval in time is equally likely for scission to occur. This also gives us now an estimate for the lifetime of a virus based on the growth rate and the length distribution. And this suggests that for a filamentous virus, a characteristic lifetime would be about 20 to 30 minutes for its assembly to take place. Okay, so this is a way that we can start to make progress on understanding the variation in morphology that's observed in filamentous viruses. But we can also now use some of these systems to quantify the amount of protein that's present in individual viruses. In particular, for any of the proteins that I described earlier, where we're able to attach a fluorophore, we can now look at viral populations and measure how much of each of those proteins is present in an individual virus. So one way that we can apply this is we can start to now get an estimate of the particles that a cell, after it's been infected, is shedding, how many of them have been successful in packaging a genome. So this is obviously something that would be favorable from the perspective of a virus, since anything that doesn't have its genetic material is going to be unable to replicate. And so by looking at fluorescent nucleoprotein as a marker for the viral genome, we can get an estimate that about 80% of the particles that are being shed that have markers of other viral proteins on them have incorporated at least some subset of the viral genome. So it's a relatively efficient process. We can also now look at the distributions of individual membrane proteins. And so we can measure distributions of HA, NA, and M2 on the surface of viruses. And if we compare this to measurements that people have made using biochemical methods, the relative abundance of these proteins is consistent with what people have seen for decades now in terms of HA being enriched by about 5-fold over NA, and then M2 being reduced relative to neuraminidase. But because we now have access to information at the level of individual viruses, we can also start to look at covariation. So how does having a certain amount of one protein influence the probability that you'll have a certain amount of another protein? And for this now, I want to go back to what I mentioned in the introduction of these two particular proteins that have competing activities, HA and NA. So if we instead look at instead of looking at the distributions of HA or NA in isolation, if we look at the ratio of these two things, instead of a monomodal distribution, what emerges is a bimodal distribution. And so what I mean by that is that there's one subset of the population that appears to be relatively high in neuraminidase and relatively low in hemagglutinin, and then another set of the population that's higher in hemagglutinin and lower in neuraminidase. So essentially another way to look at this that I'll be returning to throughout the following slides is this sort of two-dimensional plot here where HA per virus is represented on one axis and neuraminidase intensity is on another axis. And you can see these two distinct populations that have emerged. I haven't looked at that yet, but that's a great idea. So I'll describe in a little bit some of the work to look at how different members of the cellular population are contributing to this. But something that would be really interesting to look at is if some of these are early versus late or something like that. So this observation was unexpected, and so we wanted to try and understand what might be giving rise to this bimodal distribution. So the first hypothesis that we had was that it could be due to variations in cell-cell expression levels. So for example, if one cell is infected and is making relatively high amounts of HA but lower amounts of NA, it could contribute to one set of the one set of that two-dimensional parameter space that I was showing, while another cell with a different expression profile could be contributing to another part of that parameter space. So in order to test if this might be the case, we can do an experiment where we take a monolayer of cells, we infect them, and then we label neuraminidase and hemagglutinin on the surface of those cells. We can then segment these images and calculate the average intensity of the two different proteins on a per-cell basis. So if we do this and we plot it in terms of the average HA and the average NA on a per cell, we get something that's very, very widely distributed. But if we look at the ratio, it doesn't appear to be bimodal. So we get a widely varying but monomodal distribution. So this doesn't seem to be quite sufficient to describe the variation that we're seeing at the level of viral populations. So another hypothesis could be that it could be variation at the level of individual cells. And one model for this could be if there's, for example, variation in the cell surface distribution of the two different proteins. So for example, if the budding process is sampling from a demixed distribution of two proteins on the surface, it could potentially give rise to something similar to what we're seeing. So in order to test this, we designed an experiment where essentially we take a monolayer of cells, and these cells are polarized, so we can essentially infect them at a very low multiplicity of infection, so that there's good spatial separation between neighboring infected cells. We can then flip them over so that their apical surface where the virus is emerging is a few tens of microns above a functionalized cover slip. We can continue to feed these cells through their basolateral surface. And then we can essentially get a fingerprint for viruses that were shed by a single cell by looking at their spatial distribution on the cover slip. And then we can label and analyze them in the same way that I was describing previously. So if we do this, this is sort of characteristic of what we see. So as you might expect, the smaller spherical particles diffuse more rapidly and have a larger footprint, while the longer filamentous ones tend to settle more or less directly down below the cell that they came from. But if we analyze this data that came from a single infected cell, infected with a single virus, we start to see the emergence of these two different populations that I was talking about previously. So this suggests that essentially all of the variation, or a significant portion of the variation that we're seeing in virus that's collected from a population of infected cells, is essentially emerging from an individual infected cell. So an individual infected cell is capable of producing most of the variation that you would see in, across a population in terms of viral phenotype. So this, as I mentioned, one way that one model that would potentially account for this is if the distributions of these proteins on the surface is heterogeneous and somewhat patchy. And so we can start to get an indication that this might be the case if we again look back at filamentous virions, where the virus itself is stretched out to the point where we can start to resolve features at subviral level. And so in particular, if we look at the distributions of HA and NA on these two filamentous virions, so NA is in red and HA is in blue, you can see that the NA distribution is somewhat punctate along the surface. And based on the intensity of these molecules, we're not looking at individual proteins here, but rather clusters that correspond to something on the order of tens to up to potentially 100 proteins. So these are relatively large clusters. So there's many different ways that you can go about modeling a non-ideal mixture such as this. So one way that you can do it is, for example, a two-dimensional easing model. So here, if we model distributions of HA and NA in this way and then sample them and bin them essentially by size, we can start to recapitulate, at least superficially, some of the features that we see in our measurements. So I don't want to overstate the significance of this, because there's many different ways that you can model a non-ideal mixture, and this is just one of them. But I want to highlight the fact that this is now the sort of model that we can go about more rigorously testing using some of the measurements that we're able to make, with the ultimate goal of getting a deeper physical understanding of why the proteins might be partially segregated on the surface of the cell, and how that might translate into the composition of viruses as they're being built. Okay, with this I'd like to kind of summarize this first part of the talk. So far I've described a technique that we can use now to do site-specific labeling of viral proteins in a non-disruptive way, and how this allows us to look at the assembly process and the architecture of released virions. So in particular, we find that the virus morphology is consistent with the constant rate of growth and scission occurring at a uniform rate. And also we find that looking at covariation in HA and NA on the surface of the virus is consistent with partial demixing of these proteins on the surface of the cell. So what I'd like to now shift gears to is to trying to understand what the significance of this might be. So why might it be in a virus's advantage to assemble in this particular way? And in order to do this, I want to invoke kind of a case study in virus adhesion through multivalent interactions. Okay, so far I've been talking about morphology and composition as two separate things. But there's at least one way in which they're intimately related, and that's that both of them influence the ability of a virus to bind to a host cell. So one way that you could think about this is if you imagine the interface between the viral membrane and the cellular membrane, you could characterize it by the number of HA molecules that are in the viral membrane and the number of sialic acid residues, the viral receptor, on the cellular side. So essentially, using a statistical model that was developed in David Shah's group last year, we can essentially calculate the binding affinity based on these numbers, in addition to the equilibrium binding affinity between a single molecule of HA and a single residue of sialic acid. So this effective binding constant, if we plot it as a function of the number of HAs and the number of sialic acids, gives us something like this. So sort of like a phase diagram where there's an abrupt transition between virus being released with very high probability or virus being bound with very high probability. And so you can imagine now that composition and morphology provide different ways that you can explore this parameter space. So for example, if you imagine a smaller spherical virus versus a longer filamentous one, the interface between the virus and cell would be different in these different cases. And so it would be characterized by different numbers of HAs and sialic acids. And so essentially by changing morphology, you're sort of exploring this parameter space along a diagonal. Alternatively, if you keep morphology constant but you vary the composition of a virus, you can explore along the vertical axis. So the amount of HA is what's changing in this particular case. So the idea of kind of varying your binding affinity is something that a virus could do through morphology and composition, but it's something that it also needs to be able to do temporally. So it needs to be able to bind to a cell to initiate infection, but it needs to be able to escape after it's succeeded in that. So I'll walk through what I mean here. So for example, if you imagine an uninfected cell and its first encounter with a virus, the virus wants to be able to bind with a high probability or have a high binding affinity. So this would put it here. Similarly, the cell would have its full complement of sialic acid at this time point. As the infection progresses, however, neuraminidase is expressed and secreted to the surface of the cell, where it starts to destroy sialic acid. So essentially, that means that you're being pushed in this direction over the course of the infection. So this virus was able to successfully bind, and as a result, the amount of sialic acid is being reduced. As the infection progresses, the infected cell starts to produce new viral particles, and these are now in a regime down somewhere in here. So they're able to detach and escape to spread infection. If, however, we imagine inhibiting neuraminidase through some means, we could potentially push things back into this regime where there's still a significant complement of sialic acid left on the surface of the cell, and this could inhibit virus detachment after the initial infection. But similarly, if some subset of the population had, for example, lower HA content, potentially it would now be better suited to escape. So the idea of modulating the amount of sialic acid through the inhibition of neuraminidase is not just an academic exercise that I'm walking through here, but this is how the most widely used anti-influenza drugs operate. So for example, this is the molecular structure of cell surface sialic acid in the neuraminidase transition state. And these are the two most widely used anti-influenza drugs. So zanamivir is sold under the trade name of Relenza, and oseltamivir is sold under the trade name of Tamiflu. So essentially, we can now use these as tools to modulate the amount of neuraminidase activity and see what effect it has on viral populations. But before we go about doing this, we want to characterize the potency of oseltamivir or Tamiflu in our in vitro system. So we can do this by infecting cells at a multiplicity of infection of about 0.1. adding the active form of this drug, so oseltamivir carboxylate, and then collecting the virus at 24 hours post-infection. And so if we do this, we find that there's a regime where essentially a 10-fold increase in the concentration of Tamiflu essentially decreases the number of shed particles by about 10-fold. And we can see sort of a similar result by just looking at these cells with a bright field microscopy. So in the absence of treatment, the cells have rounded up and are these high contrast objects here that are dead. But at relatively high doses of the inhibitor, the cells are doing reasonably okay at managing the infection. So we can also now try and get an estimate for what a physiological working concentration of oseltamivir might be. So based on the literature, we would expect for 1 to 2 daily doses of about 75 milligrams, the bioavailability and peak concentration of oseltamivir in the sinuses would be somewhere in the vicinity of 100 nanomolar to 1 micromolar. So if we look at our result here, this puts us in a regime where we're strongly inhibiting the amount of virus that's being released. So this is a log scale. So these are about attenuated about 100 to 1,000-fold over the case with no inhibitor. But there's still a significant a significant fraction of the viral population that's being released. So we can now return to the imaging experiments that I described earlier and now look, in addition to HA and NA, how much sialic acid is left on the surface of the cell in the presence or absence of neuraminidase inhibition. And so if we do this by staining for sialic acid using a fluorescently labeled lectin, so a protein that binds to sialic acid in the particular type of linkage that the virus we're working with prefers, we can get measurements for the amount of sialic acid that's remaining in cells that have been infected, cells that are uninfected, or cells that have been infected and then treated with the inhibitor. And what we find when we do this is that in the absence of neuraminidase inhibition, infected cells do a very efficient job of cleaving sialic acid. So essentially, the signal that we're seeing here is at background levels. In contrast, cells that have been infected and then treated with, at the lower end of the physiological dose of oseltamivir, have about 50% of the sialic acid content that uninfected cells have. So there's about half of the sialic acid is left behind in this case. Okay, so we can now plug some of these numbers back into the statistical model that I described previously, combined with some physiological estimates for what some of these numbers might be. So for the number of sialic acids at the virus-cell interface, we estimate from the literature about 2,000. For the number of HAs at the virus-cell interface, somewhere on the order of 25 to 75. And a typical binding affinity between a single HA molecule and a single sialic acid would be on the order of a millimolar. So the individual interaction is actually quite weak. It's only through the multivalency of it that the viruses are able to bind quite tightly. So now if we consider a case where about 50% of the sialic acid is being left on the surface of the cell, this now puts us in a regime where we're kind of close to the threshold between viruses being able to detach and viruses being essentially sequestered on the surface of the infected cell. So this gives us some confidence that we can now use our experiments to measure shifts that might be relevant for what's going on with a physiological dose of the neuraminidase inhibitor. So the way that we can do this experiment is essentially infect cells at a relatively high multiplicity of infection. So we're looking at just a single round of replication here. About 16 hours afterwards, so I should say we can either treat or not treat with oseltamivir, the neuraminidase inhibitor. We can collect the virus that comes off at about 16 hours post-infection. We can then add an exogenous sialidase to cleave virus that had stayed bound to the surface of these cells. And then we can collect what comes off there and look at that as a separate population. So when we do this experiment, in the absence of any inhibitor, we see something similar to what I described previously. So we have these two different populations. About 30% are in the higher NA, lower HA pool, and about 70% in the higher HA, lower NA pool. In the presence of about 100 nanomolar of the oseltamivir, the neuraminidase inhibitor, we see two things that are somewhat subtle that start to happen. So first, we get an increase in the higher NA fraction, and we also get an overall decrease in the amount of the HA intensity that we're labeling per virus. So a shift from about 3,000 to 4,000 counts down closer to about 2,000 counts. And finally, if we then collect and image the viruses that are released only after we've supplemented an exogenous or a supplemental cialidase, we recover the population that was higher with hemagglutinin, and then not very much comes off in this Na-rich pool. So this is sort of consistent with what you would probably intuitively guess might happen given the nature of the inhibition that we're using here. But what remains unclear based on this is precisely what is going on. So for example, are the changes that we're seeing due to changes in morphology, changes in composition, or some combination of the two things? So to try and get at the question of morphology, we can image the viruses directly and look at their length distributions. And so what we find in the absence of the inhibitor, we get our characteristic mix of filamentous particles that are several microns long, mixed in with smaller ones that are below the diffraction limit. But as we increase the dosage of oseltamivir through this physiological range, essentially we're losing the release of the filamentous population until by the time we're at the higher end of the physiological dose, essentially we don't see any filamentous particles being released. So there's two potential things that could be going on here that could explain what we're seeing. So in one case, as I mentioned, increasing the contact area between a virus and a cell increases the binding affinity. So this could just mean that longer, more filamentous particles are preferentially being sequestered when there's a lot of sialic acid left on the cell. But the other mechanism that could be at play refers back to what I was referring to earlier about the growth of these viruses. So if we imagine the growth of a viral particle in the presence of interactions between neuraminidase on the surface of the virus and sialic acid on the surface of the cell, or I'm sorry, hemagglutinin on the surface of the virus and sialic acid on the surface of the cell, this could essentially act as a physical restraint on the budding process itself. So we could actually be getting an active adaptation in some sense, in that inhibition is actually making the viral particles that are being produced shorter, in addition to biasing which ones are able to detach. So far, everything that I've been discussing is in the context of some sort of human intervention. So we're using a synthetic chemical to inhibit the activity of neuraminidase to look at the effect that it has on a population of viruses. But I think there exists the possibility that natural variation in the growth environment of the virus could have a similar sort of effect. And so one example of where this might potentially be relevant is the temperature in which a virus is growing. So for example, if we compare the characteristic temperature of the upper respiratory tract and the lower respiratory tract in humans, there's about a 4 degrees C difference in these two different growth environments. And this temperature range that's relevant for influenza viruses increases even more so if we consider the avian enteric tract where it's replicating in birds. So this sets up a relatively straightforward experiment where we can now do. We can grow cells and viruses at different temperature and look at what's released. And when we do this experiment, what we find is, again, at 37C, we see something similar to what I showed previously. But in the case where the virus is now grown at 33 degrees, we see something that superficially resembles what we saw with the neuraminidase inhibitor, but is even potentially more dramatic in terms of shift from occupancy down here to this population up here. So obviously, when we're changing the temperature in which very complicated biological processes are taking place, a lot of things are likely contributing. And this makes it very difficult to say mechanistically what might be going on here. But I think, nonetheless, it provides evidence that variation in just the natural growth environment of a virus could be influencing the phenotype of the types of particles that are being shed. And this is something that could be quite general. So with that, I want to propose the idea that phenotypic variation in a virus, rather than being a liability, could essentially be an evolutionary hedge against a complex and unpredictable environment. And so while this might not be the most surprising or controversial statement, I think it's nonetheless interesting to consider why some viruses would favor a strategy where their progeny are extremely homogeneous, while others would take a different tack and produce progeny that are highly variable. But in the context of influenza virus, I think one potential model or way that we can think about this is that even in a genetically more uniform population, phenotypic variation could assure that some subset of that population is well suited for the environment that it finds itself. This subset could then go on to infect the next cell and essentially buy the virus time to accumulate the genetic mutations that would be necessary for a more long-term adaptive response to whatever challenges it's facing in the environment. And this could essentially be an interaction between phenotypic variation and genotypic variation that I think is interesting to consider. And finally, the idea that a virus might need to hedge against unpredictability and complexity in its environment, I think, is particularly interesting to think about once you start to consider exactly what sort of environment these viruses are emerging into. So this is just two confocal slices of respiratory epithelial cells that I've differentiated in an air-liquid interface. And what we're seeing here, so this is through the cell body, and this is at the top surface of the cell. So this is the surface from which the virus is emerging. And labeled in green here is MUC5AC, one of the major gel-forming mucins. And you can see on the top surface of this gel, so the first thing that a virus would encounter is essentially a thick, microns thick field of mucus that presents both a physical and a chemical barrier to the spread of infection. I say chemical because it's particularly rich in sialic acid. And so some of the questions that I was addressing about HA-NA balance just in terms of escape from cells are also quite relevant in terms of looking at the ability of a virus to spread in this type of chemical environment. So I think if we take a step back and take as our goal kind of understanding more comprehensively what the molecular and physical basis for infectious disease is, this is certainly a very significant challenge. But it's one that I'd like to propose. We have now some tools in place that will allow us to make progress in this direction. So in particular, by using chemical tools like some of the ones that I've described here today to study the assembly process and the organization of viruses in more detail, I think it's possible to gain new insights that complement all the fantastic work that's been done using biochemistry and electron microscopy. So this just provides us a new dimension to do these sorts of studies. In addition to this, I think an interesting direction to consider is, rather than studying the virus as it exists in nature, can we try and gain insight into the process of assembly by trying to build it from the bottom up? And what I mean by this is, can we take purified viral proteins, for example, and model lipid membranes and study their interactions with each other to gain some insight into what's taking place inside of an infected cell? So in work that I didn't have time to talk about today, I've done this using the matrix protein 1 and model lipid membranes. And what we've essentially found in this work is that if we get the lipid composition just right, we can find conditions where the matrix protein labeled in green here is able to bind to deform the membrane and eventually pinch off into these luminal virus-like particles that I'm highlighting here. So this is obviously a severe oversimplification of an actual virus. But it provides a simplified platform for now understanding some of the interactions that might be underlying the assembly reaction. It also provides us a foundation to start adding in additional complexity, in particular looking at how some of the membrane proteins from the virus might be contributing to assembly, and how they might be interacting with each other to bias how they're incorporated into viruses, and potentially get at some of the questions that I discussed earlier about the balance between HA and NA. I think in addition to trying to understand the assembly process, I think better models to understand how the virus is transmitting throughout its environment and navigating barriers like host mucus are also important. And so I think the ability to design in vitro systems that better model host physiology using microfluidic devices is a promising direction to pursue. And ultimately, my hope is that through the insights that we gain into assembly and transmission through some of these efforts, we can start to understand the virus better and provide ourselves with platforms that could potentially allow us to identify new small molecule inhibitors of any part of the replication cycle, and also provide us insights that could be useful for repurposing the virus or re-engineering it to tweak its function. So for example, that it could target malignant cells and leave healthy tissues alone. So with that, I want to wrap up. I want to thank all of you for your attention. I want to thank my home lab at Berkeley, led by Dan Fletcher. It's a very crowded but fun environment to work. I want to thank my sources of funding and you for coming to hear me. And any questions that you have, I'd be happy to take.